So we've talked about this concept of these nucleotides getting formed, and we said the nucleotides were basically this base, and then it's hooked up to a sugar that's ribose that we now find out, and then that's hooked up to a phosphate group, and all three of these components make this thing called a nucleotide, and the nucleotides begin to hook up with each other to make these nucleic acids, and these nucleic acids are what we call DNA and RNA. So we need to take a look at how the actual hookup happens. And what we say when the hookup, when this polymer begins to get formed, is that the polymer is connected in a 5' and a 3' end. So we need to take a look at what 5' and 3' really mean, because if we don't know where that's coming from, then we don't really understand the concept if they write it down for us. So what is the 3'? What is the 5'? Where are those positions located? And that will allow us to start getting an idea of how these nucleic or nucleotides make nucleic acids. So the 3' and the 5' are going to be regarded as the carbons on the sugar, the carbons on the pentose, the ribose. That is what we're after. So that's what the 3' and 5' represent. You have to imagine the primes meaning basically the other molecule. So carbons 1, 2, 3, 4, and 5, and so on, imagine those at the bases, up at the top. And then the bases are attached to the sugar, and those are carbons 1, 2, 3, 4, 5 in the sugar. But because they're different 1, 2, 3, 4, 5s, we put a prime on those. If that was hooked up to a third molecule that had other carbons, and those carbons were carbon 1, carbon 2, carbon 3, carbon 4, we could call those carbon double primes. So 1 double prime, 2 double prime, 3 double prime, and so on. Hopefully you get the picture. So that's what we're doing. We're talking about 3' and 5', meaning the third carbon in the sugar and the fifth carbon in the sugar. So primes represent the ribose. And when we talk about hooking up in the 3' and 5' orientation, we're looking on the third and the fifth carbon of the ribose sugar. So again, carbon 1 is here, carbon 2 is here, carbon 3 is there, something special is going to happen here. Carbon 4 is there, carbon 5 is there, and again, something special is going to happen there as well. So what exactly goes on in these locations? What goes on at the 3'? What goes on at the 5'? You know, these are the special points on the sugar that begin to hook up all of these nucleotides in a row to make the nucleic acid. So at the 3', the OH group is going to leave. So the OH group is going to get removed, and the phosphate on another nucleotide takes that position. So that phosphate group is actually attached to the 5' carbon of the nucleotide. And that's why we call this a 3' to 5' hookup, because at the 3' location, that OH group is removed, and that OH group is replaced by the phosphate on another nucleotide group at the fifth carbon. We call that a phosphodiester bond. You know why it's called a phospho? Because a phosphorus is included, and the diester portion is because it kind of makes an ester in a way. So if you take a look at kind of the basic fundamental hookup of the DNA backbone, this is what you're going to see. So here we see all of the bases that are kind of pointed off, A, G, C, and U. So we're talking about uracil here, meaning we're probably referring to RNA. I know it's RNA because at my second position, I see an OH group that is there. This is not the deoxy form, so it is not the DNA form. This is the RNA form. So a base is attached to my sugar. And this sugar is hooked up to another one, which is hooked up to another one, which is hooked up to another one through a phosphodiester bond. So ester group, if that was a carbon, C double bond O and O. So a C double bond O and O, that's kind of one ester. And then a C double bond O and an O over here, think of that as kind of a second ester. So diester is what we call that. So phosphodiester bond. That's the hookup spots to hook all of the sugars up, which are your chain links to make the entire chain. Okay, so here on the fifth carbon, 5 prime, that is where this phosphate would normally be. This thing begins to make a polymer by targeting another base, or another sugar, and that removes the OH group, and the phosphate is the connector piece. So here they're actually drawing it out for you. So imagine this is where it starts, right, or ends, depending on how you view it. Is it half full or half empty? So here's the 3 prime end. There's the 5 prime end. At the 5 prime carbon, that's where my phosphate is at. This phosphate hooks up to a 3 prime carbon on another sugar. That 5 prime carbon has a phosphate. That hooks up to another 3 prime sugar. That one has a phosphate. That hooks up to another 3 prime sugar, and the chain continues on and on and on for maybe billions of base pairs, because again, these molecules are large enough for you to actually view them and see them. So that is what we refer to as the phosphodiester bond. It's the phosphate group that is the glue in a way that holds the sugar backbone together. And if we destroy that phosphodiester bond, then we can get these things to break off into fragments and pieces and start isolating one base at a time if we need to do that. So this is the structure of the DNA backbone. It's sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. Can I make it any clearer? And off of these sugars, they have bases. And these bases are my A, T, G's, and C's. And if it's RNA, it's not A and T, it's A and U. So A, U, G, and C. So that's the structure of the DNA molecule itself. Okay, so there is one representation of a DNA backbone. Here is another shorthanded representation of the DNA or the RNA foundation. And the B is representing the base, and then we see the 5-membered ring, which is the sugar. Notice there's no OHs drawn on them at all. And then we see the phosphorus that connects all of the pieces together. So again, phosphorus, sugar, phosphorus, sugar, phosphorus, sugar, phosphorus, sugar, phosphorus, sugar. And these phosphorus are hooking up the sugars on the 5 prime and the 3 prime carbons. So that is another representation of the DNA molecule itself, or the RNA, depending on what version I have. If I take a look at this structure and I asked you if this was DNA or RNA, and I didn't tell you that the structure left out all of the H's, all of the OH's, and everything else in between, you would look at these and say, well, you know, I'm probably going to assume that this is DNA just because they don't specifically draw the OH group there. So because there's no OH group there, then it's probably the RNA form or the DNA form. No OH, deoxified, this is the DNA. Not only that, but something that I don't like about this structure even more is that they try to simplify it too much, and we understand that this is a sugar here, and this needs to be an oxygen, not a carbon. But again, they're doing this because they want to simplify it as much as possible, and they just want you to kind of focus on the foundation, and that's really all that they want to do. So they're leaving out, again, all of the oxygens, all of the hydrogens, all of the unnecessary portions that make make up the backbone. And they just want you to take a look at the hookups. Where do the hookups happen and how do they form? So again, 5 prime, 3 prime, and that connector piece is the phosphorus. That's what brings both pieces together. So there's another representation of what we need to do. On a test, this could be a question. And I could ask you, basically, draw the foundation of DNA or RNA. And this is what I'm after. So you don't have to go in specifics and you don't have to go into details. You could replicate that diagram, and over to the side you can put P represents phosphorus and the B represents the base, and in parentheses put ATGC. And then this proves to me that you kind of understand how the DNA and RNA is made as far as the backbone goes. So this is okay with me if this is what you want to replicate on a test for a test question. I don't need exact structures with all of the phosphorus, with all of the oxygens, with all of the hydrogens, with the stereochemistry involved. I could do that if I want you to hate me, but really we're at the end of the semester. Why go through more than what we have to go through? This proves to me that you understand the basics of the DNA structure itself. Now, if you really wanted to be nitpicky and go into detail, this is something that you could draw. And now you kind of see the big picture here. You understand that this can become as complicated as you want it to become. So in this diagram, what you're seeing is that the bases are all drawn out. So here they've fully drawn all of my purines and pyrimidines, my ATs and Gs and Cs. And then I'm showing where that's connected to the ribose portion. And this ribo portion on my 3rd carbon and 5th carbon, that's where I see my phosphorus, and that's where the hookups are. They're also showing you the negatives that are happening on the phosphates, and we call that the phosphate deoxyribose backbone because this is DNA. I don't really see an OH group that's here. And not only that, but they're showing you the complementary strand over here as well. And in the complementary strand, they're drawing the bases out as well, and then they're showing you with dotted lines all of the hydrogen bonds that happen in between the bases. So this is really a full picture of a double-stranded DNA. One phosphate deoxyribose backbone with the bases, the complementary strand with the bases, and how those bases pair up into these couples that come together that help these two strands hook up and stay stable. So just another representation of the DNA strand itself. Okay, so by convention, we always write the order of the strand in the 5' to the left and the 3' on the right. So if I go up and take a look at this structure again, the 5' is where I start. This is called the 5' end, and that's my phosphate. So my phosphate's attached to the 5th carbon. By convention, just because this is what we said that we're going to do, we always start writing the order of the base pairs on this end. So this is my starting point. So you got to be careful. I want to make it very clear, and I want to put a system in place when I begin to report the order of the base pairs. And we look at the phosphate that's attached to the 5th carbon. That is my starting point. So with the starting point, I then say this is base number 1, I write that first. This is base number 2, I write that one next. This is base number 3, I write that one next. This is base number 4, I write that one next. So that is what we mean by writing them in 5' to 3' ends. That is the order in which we go. We only write down the letters A, C, T, U, and G depending on what we have, and we leave out everything else. Sometimes the ending gets a P attached to the symbol, which means ending pair. That means it's a full stop. This is truly where it ends, and sometimes in the beginning we put D if we're talking about DNA. So in the next video we'll continue on with this convention idea, and we will talk about how we write write things down and how we interpret that information.